light room. Might as well. And then
Sorry, I don't look very glamorous today. I'm just on my
it's totally fine. No, I mean, the reason why I always, like, my hair is always wet in the beginning is because I always take a shower because I work out before it. So I rapidly comb my hair in the hopes that I look okay.
Well, I normally have pretty like wavy or curly hair, but it gets uncurly after I sleep on it. So most of the time, especially now with COVID, and I haven't gone to get a haircut, normally like it shorter. Now it's long.
Yeah.
Because I normally just put it in a bun and just leave it up. But
Right.
But because I had my seminar today and I had to kind of look professional, I straightened my hair and whatever. But I took off my top like professional attire and just moved to sweatshirt.
Right.
Because I was like, once that's
Of course.
over.
I mean, I'm wearing a Pokemon, like, Solar Gym shirt, so, like, we're all professional here.
Yeah, I'm wearing a sweatshirt with a
that's
teddy bear.
that's
Like,
a great. Oh my god, that's a great shirt. And your phone autocorrects the duck, but it's okay. Sponge, so I can soak out some facts equal duck s facts. So I'm just reading what chat's saying.
Oh.
Name for the minions, Jack's Facts as well. Someone in Jack's Packs. Someone suggested Jack's Packs. I thought that was a good one for the crew. All right. So, just kind of an introduction for everyone here and for those later. This is Dr. Allie Graham. She is a professor at the University of Utah.
No, a postdoc.
Postdoc. Oh, I thought professor. Your site. You have so much research. I thought you were already a professor.
No, I'm applying.
That's awesome.
Nothing yet, so I'm just a lowly postdoc.
Yeah. For those who don't know, a postdoc is when after you get your PhD, you then go and work either in the lab you were in the whole time or in a different lab, and you're kind of the rung underneath the primary investigator, and you usually help them with a lot of their research, and you conduct a lot of your own research just to kind of expand your repertoire so universities will take you.
The role of postdocs it didn't used to be so common, but with there being kind of less and less faculty positions and like more people, so it becomes kind of like a way for you to stick around a bit longer, but it is a way you're supposed to become more independent. You're not having to also take classes or anything like that. And you're also potentially training or helping train like graduate students and undergrads. And then the idea is you're supposed to kind of be coming up with your own research program in theory so that you can take it with you to form your own lab, at least in the sciences. It's obviously postdoc is not really like much of a thing outside of the sciences.
Right.
Yeah, they just get their PhD and they just go straight into being a faculty.
Yeah.
Like in sociology or English or whatever.
Those lucky dogs.
Right.
Yeah. Also, Chad, I just side note, if the stream is kind of being a little choppy, let me know so that I can try to adjust. I looked at some of your. So actually, one of the first questions is, what got you into animal behavior? Like, what made you want, and genetics, like, what made you want to go into animal sciences?
Well, it's one of those things. I'm always fascinated to hear people's stories because I feel like when you see, experiments or being talked about in textbooks, or maybe you hear even seminars, it kind of gives you this false sense that everybody just had it all figured out. I mean, maybe some people like were very driven and it's like it just fell into place for them. But for most people, they take this long, circuitous route to kind of get where they are. So I went to a very small women's college for my undergrad, and we didn't have any research programs of any sort. I helped TA and things, but I also had to work during the summer to help pay for school and stuff. And so I didn't really know what like research was. I just knew that I liked science, and I had always been interested in genetics. Though, like not from a medical standpoint specifically. I don't think I have that good of a like bedside manner. I'm a little prickly sometimes, but, so I knew I wanted to kind of do that. I just didn't know what that meant, right? Kind of coming from a first gen family, you know, I didn't know. So it was just kind of like, so I didn't think I was ready for a PhD, and I kind of was encouraged by some people that were like, well, you know, at the very least, you know, like if you're in a PhD program and you don't like it, then, you know, maybe like you come out of it with experience. But, you know, with a master's, at the very least, if you don't like it, you just like suck it up for another year and like you're out kind of thing. So like, so that's kind of how I ended up. I did my master's first.
Yes, at UNC Greensboro.
And yes, at UNC Greensboro. And he took like a really big chance on me because I didn't have any lab experience besides the ones that we had during my undergrad, you know, in classes. And it was in an evolutionary genomics lab in honeybee research. And so looking at the eusocial evolution of honeybees. So obviously a lot of Hymenopteran species have this really complex caste system.
And can I pause you for a second? Can you explain what Hymenopterans are? Like for I think I know what they are, and eusocial too. If you could break down that for everyone, that'd be great.
So Hymenopterans are kind of like the group of insects or the order of insects for like bees and like wasps and let's see, hold on. It's been so long since I've taken entomology. Let's see, Hymenopterae. I just want to double check and make sure I'm not telling you.
I always Google too.
So yeah, it's wasps, bees, and ants. That's what I thought. So like there's a common feature for a lot of those groups where they have these kind of complex caste systems. They range in terms of like they're being really discrete, you know, workers, drones, queen, or like there's some wasp species which have like a really kind of fluid, you know, like just pecking order, but not like discrete. You know, like these can reproduce and these can't reproduce.
Right.
And so eusocial is the description about, I think technically it just describes that type of caste system. It's been so long since I've talked about eusociality for my master's. It's just social organization where like you end up having a single female reproductive member, and then the rest are non-reproductive, except for the males. And obviously the drones, they don't stick around for very long. They get kicked out, and then they mate, and then they die, kind of thing. So it really is a female-centric set of systems. So the project I worked on was to do with, it was kind of still in the news at the time. You guys remember talking about like Africanized honeybees and like them, that was the kind of bugbear associated with like, oh no, we're going to be invaded by these like horrible, you know, stinging things. And now it's, what was the fad there for a bit?
The Japanese,
those wasps, like giant wasps.
Japanese giant hornets.
Or whatever.
Terrifying.
Yeah, the hornets. So there was, you know, there's differences in behavior with Africanized honeybees, even though they're technically like the same species, and they're not different in any particular genetic way that they knew about, but they're more aggressive, and they're kind of ovary size. The reproductive organs were bigger and whatnot. So they, in conjunction with some people at Arizona State, I believe, who had like the certificate, I think, to technically to be allowed to handle and breed Africanized honeybees.
Wow.
They did like complicated crosses. So I never participated in that.
Right.
I had all my samples in like the freezer, which is like a common thing for a lot of people that mostly do computational work now, which is what I do. So it was trying to see if there was, so they did these crosses between like European and Africanized honeybees as a way to try and parse out the specific genetic regions or in the genome that might be associated with both ovary size and kind of trying to connect it back with behavior because of all this other previous work that had been done. So, so I had embarked on like a really complicated project that was like at the very end of a whole bunch of work. And so it was very stressful.
I looked at your master's thesis, at least the beginning part of it, and the preview was 24 pages long. I'm betting it must have like 50 to 60 or maybe 100.
Oh, you for my master's dissertation, or
I think I read that one because that was about the eusociality of honeybees, right?
Yeah, that was like 100 pages, and it was just one chapter, and it was I ended up publishing publishing a paper on it.
That's so cool.
Yeah, and so, but it was a lot, and I kind of wasn't as excited about it, and I was really burnt out, and so there was, so I ended up working as a technician in a Drosophila lab upstairs, in a lab that I went to all the time to use some of their machinery, and then I moved. That's where I moved
can I pause you
afterward.
Right. Can I pause you real fast? Can you? What is a Drosophila for?
Oh, okay. So Drosophila is like the genus name for fruit flies. They're not. I think there's technically like technical fruit flies, which are a different species, but these are the model system that are called that are used in the sciences that have so many resources and that have been used since the 1800s, mid 1800s, and later than that. Like, so they're a lot of what we know about genetics is because we've been able to kind of manipulate these fruit flies. And it's, you may have heard it because a lot of times politicians will be like, oh, why are we funding research on, like fruit flies or whatever? But, there's useful tools for things. And so there was there was a lab upstairs that worked on Drosophila, like looking at how different species are kind of like created, how they evolve, and doing genomic work. And then I moved into another lab, and that's why I moved to Duke to work in another Drosophila lab, and a more prominent evolutionary biologist, Mohammed Noor, who's a huge, I think we talked about this, he's a huge Star Trek fan, and he's so nice, and now he is an advisor for the newest Star Trek series, so you'll see his name sometimes, and he gives a lot of outreach seminars about Star Trek and how, like, what the genetics of Star Trek kind of thing. So
that's so awesome.
So it was an experience. And then I taught genetics and evolution there. And then at that point, and this is like a really long, like,
no, it's great. This is great.
Science, but so from there, I decided I wanted to continue working and get my PhD. I kind of discovered I'd always been like a lifelong birder, and I love birds, and my grandma was a big birder. So like, I've always loved them. And so I kind of made a decision that I wanted to move into working on birds, like that was my like thought process at the time. And I actually, but I'd been with my now husband for such a long time, and he also wanted to get his PhD, but he had completely different interests. He does developmental biology. And so trying to find the same place, like our interest at the same location was really difficult.
Right.
We didn't really want to be apart in this, like I know a lot of people do, and it's very difficult for them, and we just didn't want to embark on that. So we ended up at the same program at the University of Miami, and we had a tumultuous time.
Really? That's
I know
so cool. My sister goes there now for her OBGYN residency.
Oh, at Miami?
Yeah.
I know they have a big medical school, and I took classes at the medical campus. And that's where my PhD advisor that I ended up staying with, he's kind of also partially at the human genetics department there too. So I had like a tumultuous time there because there was I ended up having to leave the lab that I initially like went there to go into, because he was toxic, and he wasn't good. And so that was really difficult. I spent 1.5 years in his lab, and then left and moved into the lab that I ultimately went into, which was still birds. And he had just moved from the University of Alaska, where he'd done all of his previous work. And he came to Miami because all of his work was done in South America. And so that's much closer. Right.
That does cut down like 12 hours of flying.
Yeah. So that's a whole story, because there's a whole ass thing. But ultimately it was like, I was on like a fast track to finish up and just get the hell out of there, which is like not really ideal.
I totally understand. Yeah.
So I actually finished in this is even with me like staying in the other lab, which nothing came out of that. That was a total almost waste of time, other than me like taking the required classes. I finished once I got into Kevin McCracken. He's at Miami.
That's Kevin McCracken? Wait, was that the one that you had to bounce, or was that the new guy?
No, that's the new one.
Oh, yeah.
I'm not even going to speak the old guy's name.
Right.
I don't speak his name.
Right. He who must not be named.
So Kevin is actually still my PI, and we still collaborate. We're still trying to push out stuff, and
that's awesome.
He's entertaining.
I feel like I've heard of him before. Does he also do like like mammal, like elk research, like in wolf
no,
behavior?
I think his dad does.
Was it he at like Colorado State or something?
He might. Actually, I don't know, but I feel like I know he's mentioned his dad is a professor somewhere, though I never met
Right.
him, so I'm not sure. But also, like you may have heard, Kevin's worked on ducks for a really long time, and there was like a couple of papers like that are kind of famous because of the duck penis stuff,
Which my
like,
chat is
like,
incredibly like pumped about asking, which I'm sure
yeah.
every human being on the planet asks you about duck dicks.
Yeah, I mean, so he had like a lot, like he had, I think it was like a National Geographic like printout where you
Wow.
fold out, he had this
Oh,
long,
because of the
and he put it like
spiral.
right on the window, like so, like right walking the lab. It was like I saw it every day. I've never seen any personally. So, yeah, I have very little relationship to it. Though there are people that study that because it's an interesting kind of like evolutionary arms race
right.
between male and female reproductive systems.
Right.
And it's not common. Birds don't typically have genitalia like that. They just have holes.
Yeah.
So they're like a.
Actually, something, if I may, off track for a second.
Sure.
So something that I was talking about yesterday, maybe the day before, whatever. Do you ever watch True Facts? Just random, like,
the Z Frank True Facts.
Did you ever see the?
We've been.
You ever
we've
see
been,
the?
Sorry.
Sorry, you go. I talk too much. You go.
No, I was like we've been like subscribed to his channel for forever, and I'm so glad he started back up again.
Yeah.
Yeah, and I think there was a duck episode,
Yeah.
and I think his name is at the end of that, saying like
cool.
thanks to Kevin or whatever.
So you know the ostrich video?
I mean, I've watched all of them.
Ostrich. I stretch my neck out for you song ostrich video. That's my video. Yeah. So not only is that a humble brag, but or just straight up brag, but what he doesn't show, but at the end of the video is that when the male gets off, you can see like his, I guess, a penis, and that blew my mind. So I was wanting to ask you about like how the presence of like penises in birds and such, because you're right, most of them have like cloacas, and for those who don't know, and please correct me if I'm wrong, cloacas are basically like, they're just these
openings.
Yeah, they're just openings that both you poop and you mate through. Like it's just like they do both. They do double duty. And they're just holes, and they just go like, and they kiss, and that's how most birds mate. But, yeah, because I was just like, I didn't know if you knew like ostriches had like penises and stuff, because that blew my mind when I was like filming that. I was like, what?
No, it's like very explicit. Groups of them do. I think also I know it's all that the duck I'm not sure if geese also have it. They're a related group. And, so there's kind of like these, I don't know if it's also kind of an element of like convergence. Like you've got kind of that convergence on the penis phenotype or whatever, or what. So I haven't really looked into it like that much, other than it can't help but come up as a talking point when you're talking about
always
ducks, so.
Always is. But for those who may not know, for chat and those who may listen later, the reason why ducks have penises is that it's kind of a sexual arms race between males and females, because males will physically coerce themselves, which is the fancy science term for rape, female ducks. And female ducks have evolved because they want to pick who they mate with, so they can pick basically the genetics that they like.
They don't just want to rando; they want to choose. And so, there's this arms race between one going one direction and the other going the other direction, or having these kind of like offshoot weird areas,
false pocket vaginas, basically
fake pockets. And I think so. If people are interested, I know that Patricia Brennan at Mount Holyoke University, I believe, she works on that. She works on that specific question. And she actually like did these experiments where she made like glass.
I read that paper.
And it's like to like look at, you know, the
in slow-mo. They do it in slow-mo to see how it like because
it moves around. Mm-hmm.
Yeah. What's her name again?
Patricia Brennan.
Patricia Brennan. All right, cool. But yes, duck dicks, very interesting. So in one of your, I think in the honeybees, because I was frantically reading your research site. I wanted to be prepared. Mm-hmm. You're like the first person I've ever interviewed for anything, so I'm like, I need to do it right like the first time. Later on, I'll just stop caring, but I'm not jaded yet. So in it, you were talking about kind of the end of your honeybee research. It got actually made, I think it was the Drosophila that you got into transcription factor, transcription factor research. I was wondering if you could explain kind of what transcription factors are.
Sure. So transcription factors are kind of a subset of genes that are essentially, like their name suggests, they're a factor that's associated with transcription. So I know a couple of weeks ago we had like a discussion about, you know, how, you know, DNA, RNA, protein, right, like that kind of like throughput, like your DNA is translated, is transcribed into mRNA, and mRNA is translated into protein. Right. So in order to go from DNA to RNA, you need transcription to happen, and transcription happens through transcription factors a lot. Mm-hmm. So what it does is they're proteins that will then bind to DNA, and kind of they have these ends of them that then recruit all these other factors to come in and be like, hey, like come transcribe. So it brings in the, you know, polymerase enzymes and other things to come actually go ahead and transcribe into RNA so that they can get the ball rolling into protein production, which is kind of like the ultimate goal in a way. I mean, there's you know, there's functional mRNA stuff that doesn't get translated to proteins, but right. So transcription factors are just that. But they're also, you know, proteins. Mm-hmm. And they have these most of them will have these functional domains in them that are associated with DNA binding, actually binding to the DNA, or like dimerization. So most of the time, transcription factors will have two portions that will dimerize together and then kind of like walk around the DNA in order to kind of like hold on. And then the other prime end of the protein is the thing that have what are called protein interaction domains, so that it recruits other proteins to come in. So let me, kind of if I can, notoriously difficult to work with because they're so critical, and they have this they're at this particular juncture. So a lot of people kind of it hadn't historically been something that people would work with. They'd work with other protein coding genes instead. And it's only kind of been recently that we've been able to really try and assess their like the breadth of their roles and what they're doing.
Right. If I may, for sure. Chat, I'm gonna try and draw out kind of what she explained. And I don't know if you're watching a little bit watching the stream just a little bit, just to make sure I'm not telling people wrong things.
I think technically the stream, when it comes up for me, is like behind by a few milliseconds.
Oh yeah.
I can't really, so I'm not really looking at it because it was really confusing.
Oh right, yes, that's true. That does happen all the time. All right, so here's I'm just gonna I'm going to voice it out because I always like to hear my own voice. So let's do a draw. So basically what and correct me if I'm wrong, basically what she's talking about is that your DNA is here, and it's like you have your DNA, and it goes, and when it's transcribed, it basically becomes mRNA, which is kind of half of a DNA strand, and then when that gets read, it gets into it becomes the proteins. So each of these little dots are proteins. and then the protein gets folded and everything, and bam, you've got your actual thing that functions. So what she studies is kind of like the beginning of all of this is how is kind of the process between DNA to mRNA and how that is like enhanced. Am I right?
Yeah.
So, and part of it, if I can remember correctly, is that so DNA is spiraled, and so and the tighter the coil is, the harder it is for DNA to read whatever the strand says.
Yeah.
And so,
and there's also like structural manipulations that are called like epigenetics. So like you have the chromatin that the DNA is like wrapped around, which kind of has to uncoil too. So there's several different layers in which transcription factors have to get into.
Right.
And
correct me if I'm wrong. So not only do these transcription factors at times help kind of uncoil the strand, correct? They also then I think this. I feel like I said that wrong, but this next part I feel like is correct. So when you have DNA, there's a little section in the beginning that basically is like start here to code DNA.
Yeah, I mean, there may be some transcription factors which aid in kind of uncoiling that, but the biggest thing is that because of those epigenetic.
Right.
Like if it's too tightly coiled, then it's not available for transcription. So most of the time it has to be already open-ish for them to be able to just physically like get in.
Right.
But yeah, ultimately they're like they're the things when you I remember taking intro to genetics and cell and molecular, that when they talk about general transcription factors, it can those are the first things that have to attach, and then it recruits all these other things to come in and aid it. So it just doesn't happen random, in reality. It doesn't really happen super randomly. So there's all these cascade of stuff going on. So yeah, but it physically binds because these proteins will have like really explicit motifs or like structure, patterns in the DNA that they attach to, some long, some short, that have to be present in like the upstream promoter regions of genes that promote their transcription.
Right.
Yeah.
So to summarize my attempt to summarize for everyone. So what she looks at is that at the beginning, kind of where the promoter areas and where you start the transcription of the part of the DNA you want, you don't just start with it and it just goes down willy-nilly. You need kind of blocks of proteins that kind of build on top of each other, and these are just squares, and it's on top of some other stuff that I've already drawn, but. So you build this kind of protein complex on top around the DNA that then allows the transcription of the DNA to happen at like more or less and at like different rates and such.
Yeah, it's the first step because obviously there's things that happen after that which can then lessen exactly how it's not a 1-to-1. Like once it's transcribed, it will end up being a protein. There's other things which happen and can change the ability for mRNA to actually get translated.
Yeah. So
it's the first major step.
Major step. So you can see how complicated this is, but also how important it is because you have to understand how everything else happens. This has to happen first. So you kind of like it helps to understand how everything is made in the first place. Am I correct?
Yeah. I mean, it's, just like with anything else, it progressively gets more complicated, which is why I kind of mentioned that transcription factor, like activity and evolution is only kind of really in scientific terms, kind of like recently been able to be tracked down because of that, every layer you go up, there's more and more other things that have like input. And so, I think it's in the specific transcription factor, like pathways that the one that I have consistently looked at. Yeah. So
Yeah.
I kind of fell in love with them that way because it's a challenge.
Mm-hmm.
And also because of the role and the like niche that they fill and just how important they are. And because people hadn't really looked at them before in the same way that they had looked at protein coding genes, at least in the field of evolutionary genetics. I felt like it was a great place to kind of like really try and ask and answer some more complicated questions. I wanted to move myself in the fields, I guess,
Yeah.
forward in that way. So that's kind of where I ended up.
That's awesome.
Yeah, but in terms of my interest in hypoxia, right? I mean, kind of getting back to how I ended up with my research topic like at hand, that wasn't obviously like what I intended on
It never
focusing
is.
on when I went in. So, it was like an out of necessity in a way. Like I moved into the Kevin's lab, who does high altitude duck work, and he's traditionally kind of focused on hemoglobin, the best known protein, best studied protein in like the whole world. And so, which is a clear, like hemoglobin makes sense in terms of like oxygen load and like getting oxygen where it needs to, which you'd think would be an issue for high altitude. So he's done a lot of work there. So when I started my PhD, he was like, well, he's an endowed chair, so he had all this money. So he's like, let's just go ahead and branch out. Let's think about other things that are associated with high altitude, maybe that we could test for evidence of convergent evolution or something. And so that's how it ended up with the hypoxia inducible factor pathway as like my favorite pathway in a way. But also this idea of hypoxia or low oxygen being kind of this selective pressure that was interesting, besides say like temperature or like salinity, or, there's all sorts of, you can kind of parse out the environment however you like to kind of try and answer questions. And so, but at the end of it, I kind of wanted to be able to, I was running all these computational models and doing bioinformatics, but, it would tell me the probability that something was associated with it. But I didn't really have the ability to go in vivo or like go into the system itself and probe it to like see if I disrupted that, if it would have an effect, right? Because it's hard to like apply CRISPR to like a non-model system like a duck, especially one that's in high altitude, which I had a reviewer suggest and thought was ridiculous, but I was anticipating that, which is how I ended up, even though I still love birds and I'm still kind of working in that system, I moved into systems which are more amenable to like probing the genetics like more directly to test whether or not these, computational or like running these models on the computer actually are telling us like something correct.
You had tested all of this in theory, but to like practically like adjust to actually see if what you've associated in your data, it was actually like a causal relationship.
Yeah, so that's the goal, and that's a big one. I mean, that's not something that's unique to me. That's constantly something that people are trying to do, and there's this whole what's a model system versus a non-model system, and that only comes down to like the resources available for a system. Usually the system that's been around the longest or the organism that's been around a long time, it's just had more people working on it, more money's been dumped into it, right? It's got a genome, it has like all these other things. So that's why you end up having a lot of people work mostly on these systems like zebrafish or zebra finches or Drosophila or C. elegans, the nematode, or that, because there's more ability for you to kind of go into that fine scale thing. But now with the advent of all these like CRISPR and RNAi, which was an older thing, you can actually kind of do the same thing, but in systems that didn't have traditionally all these resources. And of course, like genomes are becoming more and more available. So, so the like definition of what's model and non-model is kind of semantic, but, but obviously I also don't really, this is like as a personal level, I don't really want to go around like killing cute things that I like. And that also was a little upsetting in the first year that I was in the other lab, is I was working with hummingbirds in Costa Rica. And I was working with with a collaborator who's at a museum, and obviously museums are great resources for all sorts of things, but they had the agreement was that when I would catch some, he would have to kill them so that they could take the specimen and whatever. And it was really upsetting having to, like, I was measuring it and talking to it, and then I would hand it off, and suddenly it's like
gone.
You know, he suffocated it, because that's how you do it with hummingbirds, which is like super upsetting. So so I was like, I'd rather not I understand the importance. I just didn't really want to do it. Didn't really want to be involved in that specifically. So that also, like, all the duck species, all the duck work I did, they had been long dead.
Right.
So we were like dug into the freezer, and so we were adequately, like, kind of paying homage to their sacrifice like 10 years prior by like only going, not having to kill new things.
Nice.
Though apparently it's easy enough to do, because part of his like research materials, Kevin's, was like a shotgun and shotgun shells, because you just go out there. Like,
so you them. Just go like bird hunting.
They're super common. They're like not. Okay. They're not a threatened species. Obviously.
Well, that's good.
And they're like, so, you know, we get into that, but like these ducks in the Andean mountains live on this what's called the Altiplano Plateau. It's like the largest plateau in the world. I think technically it's maybe even larger than Tibetan Plateau. But so it's high altitude, and it has all these lakes and stuff on it, and so a lot of things like flamingos and other waterfowl live up there. And there's like no predators besides people for the most part, and so they're really, like, they're just out and about, so you can just go out there and shoot them. They're really common hunting species, and so you just go out and do that. So you have a bunch of these different duck and waterfowl species that independently colonize these really high altitude locations, because it's a niche to move into and there's not a lot bothering them. So that's also great, but also you have low oxygen, which is a problem in its own right. So that was kind of the what I was trying to address. So I had both low and high altitude populations, and I was comparing between those, but also comparing between species that had independently done the same thing, to see like just how predictable evolution was with selecting on similar molecular pathways, right? Not everything is technically like up for selection to mess with. But it has to work within a pre-existing framework, kind of like city planning, right? Like you can't just up and rip up a whole, like portion of like an area, like it's a whole thing, right? So you kind of have to work around existing structures to kind of get what you're going to get. That's not necessarily optimal. It just is it's good enough. Like most of the time they talk about evolution as
evolution optimal. Is most of the time enough.
It's just good enough. Like, so it's government work, right? So there's this, like, there's areas which aren't really, selection doesn't test upon that, or doesn't deal with, or there's mutations that don't occur in there, because if they do, like, it's lethal. Like, that's a problem. You don't
touch them. They're no-nos.
You're dead. So,
where did the mutation touch you? These are the no-no spaces.
But there's some areas. So like there's kind of these stipulations about, like, there's a landscape across the genome in which things are more or less likely to be selected on, because of these stipulations about just how important they may be during like development. Generally you don't want to mess with something, so that's important for you to like develop. So there's this kind of like give and take there. In terms of that, and there's a lot of literature kind of exploring that topic. So that's kind of where my questions about like the predictability of evolution and that convergence, right? There's that layer of do similar things pop up as being important in different animals, like humans and ducks and yaks and whatever else. So that was that.
So if I interpreted correctly, what you're going for is to see, because of how your genome has certain areas of like, you cannot alter this, you will die, and other areas that are like it's okay to adjust because of like the restrictions of how of where mutations can occur. When you put separate species into the same environment, they're going to convergently evolve similar things because it's the only parts of the genome that can be adjusted for such adaptations.
Yeah. So there's a limited, your probability that these things that can be in specific areas is not equal amongst all things, right? And it depends on what environmental factor you're like talking about in a way. There's a lot of stuff which, the pressure is that so like that most of the time if it's a lethal thing that it's messing with it, that's important for development. There's usually not that many things that do, though there are quite a bit of examples of changes that have occurred in developmental genes which have changed the phenotype or like the body plan or other things in such a way that we get these leaps in development. So and in evolution. So that's not a huge no. It just means that the probability of that happening and it being okay is much lower than like other areas where it's like, yes, it's more okay and less likely to.
The room for error for like creating something bad.
Yeah, there's always this, and them obviously oversimplifying it, but in general, that's the kind of framework there. With it, so that was a big question, right? Because there'd been a bunch of work that had come out at the time. These genome-wide association studies, they were coming out with all these new genomes that were coming out, especially like in high altitude people populations. So there's ones in Tibet and in the Andes and Ethiopia. In some level, they live at high altitude. And I don't know if you guys had heard about hearing that the Denisovan populations and interbreeding there. So there was all this kind of hubbub around the HIF pathway, hypoxia inducible factor pathway, given its role. It's like kind of explicit role.
Right. Can you explain what that what the hypoxia? What's the whole phrasing of the word?
So it's easier just to say the HIF, HIF, the HIF pathway, but it's hypoxia inducible factor pathway. So HIF. Okay. So I can give you the general rundown
that would
of
be
it.
Great.
So it's difficult, but under normoxia, so under like normal situations,
oxygen
right?
levels.
And this can happen from as a whole organism. You can technically have hypoxia in like discrete areas. Like tumors sometimes create their own hypoxic regions. And in reality, we have a layer of hypoxia, like certain tissues are more hypoxic because they just come much later and the blood gets to them later. So there's like less oxygen. Anyway,
gotcha.
So there's certain like cell lines which have to be kept at hypoxia because they're used to being like that. Anyway, so under normoxia, there's a setup to where the dimers I mentioned, the transcription factors, are what are called constitutively expressed. So they're basically always expressed. They're always on. There's always like proteins of them being made, right? However, they have repression machinery. So other proteins, which then come in, tag those proteins for degradation. Right. So they like tag them and then they're
sent out to pasture.
By, like, other proteins. Yeah. So they never get to go into the nucleus and attach and, so on, like we mentioned. Right. However, during hypoxia, so there's when oxygen becomes less prevalent in the system, it actually changes like the chemistry of the cell so that the repression machinery doesn't work anymore. So that means you don't have to wait for the machine. You don't have to wait for things to then ramp up to be expressed because then your dimers are already there, right? They were just being degraded. So they can kind of turn on a dime in terms of like their ability to affect change, which is important because
it's a quick responsive mechanism.
Yeah, so there's a couple of pathways that do a very similar setup. So then when the repression machinery is gone, that means that those dimers get the transcription factors get to go into the cell and then do what they're supposed to do. And so they become this kind of they represent the major response to oxygen change because then they attach to all these different genes. They can kind of mount this response simultaneously, right? You don't have to have one for like every single gene. It's one kind of like key for all these genes to kind of get up and go. because hypoxia is a problem in theory, because we're used to having a certain amount of oxygen. It changes our energy availability and all these other things. So it ramps up this physiological and molecular response, like that just tries to get oxygen where it needs to be. However, that's only really good over the short term. Right. So if you guys are familiar with like chronic mountain sickness or other things, like there's all this stuff that happens when people who are not used to being at high altitude go into high altitude. It affects people really differently. I've never had to go, but even my advisor, Kevin, he suffered chronic mountain sickness, and people can die from it.
Right.
So it's increased breathing rate, a lot of vessels dilating. So it's just trying to get where it needs to go. But over the long term, it can be,
it's hard on the body.
It can be debilitating. It's hard. And so if something, say, has moved into high altitude and has stayed there, they live and survive, there's this idea that response is blunted, meaning that like that response is tamped down because you don't want to have like heart disease, like, you know what I mean?
Yeah.
So that's like kind of like, well, what have they done? You know, there are natural experiments. Like, what have they done, and what do they use to combat all those issues downstream? Kind of also as a way as to like how we can kind of like co-opt them or understand how to better kind of treat certain illnesses that are associated with like, you know, low that oxygen. Are kind of inadvertently associated with the HIF pathway.
Right.
So that's I don't know if that made a lot of sense. I tried to break it down.
To me, that actually made a total sense to me.
Okay.
Well, hopefully it made sense to me. I'm going to try and say it back to you in a dumber way. So, so what it seems like is that I'm going to try and draw it out for everyone. It's drawing time. So basically,
while you do that, I'm going to go get my cord.
Yes.
In the one battery. So
yeah,
I'll be right back.
All right. I'm going to try my best. All right.
Okay.
So basically what happens is, why is this not drawing? Start drawing. There we go. So basically what she's describing is that when low oxygen, when O2 is low in the body, these proteins that kind of block the transcription factors, which that block these specific transcription factors, are no longer like stopped, and because low oxygen causes them to stop, and then they go into the nucleus and into the DNA, and the DNA has a bunch of places that helps to combat low oxygen. That in short term, it makes you able to try to like keep up the amount of oxygen that you need. So, and it's a single transcription factor, so you don't need a bunch of little ones. This single one can go to here, and this gene, and immediately start ramping up the amount of like mRNA and thus the amount of protein that is created from those. And that creates that physical response of your blood vessels increasing, your breath, your rate of breathing increases, and other things that in the short term help move oxygen through you faster and more efficiently in the short term. But these things, because of like when you like things, you can't make things for free. These adaptations come at a cost because they're energetically demanding, because these transcription factors are constantly pumping out all this response, and that's what causes issues in the long term. So if you live permanently in high altitude areas, you're going, and if you have generations living there, you're going to make adaptations that enable you to live at high altitude without this response factor. So you need to but this response factor is still running. So you need to then not only create adaptations that help you survive in high altitude, you also need to create adaptations that suppress this pathway from continuously like pumping all this out because you don't need it anymore because you've got other ways to deal with it in the long term. Is that good?
That sounds right. From what I heard as I was running around.
Cool.
Yeah. Yeah, I mean, and there's also I kind of also don't want to give the impression that obviously natural selection is always working, but in theory, these, certain animals could try and go in and invade the niche, and then that they just don't have the
ability,
a wherewithal. Technically, there's not enough mutation, or there's not enough variation within the population to that could end up being adaptive in the right context, and that they just don't, have a foothold. So for everything that we see that is living there, there's probably plenty of animals which tried and failed, right? Because
they just couldn't, they just didn't have the spontaneous mutations that adjusted the genome.
There is a lot of stochasticity, or like the fancy way of saying like random chance for all sorts of things, and so it kind of depends on mutation rate, where those mutations occur, and like I said, standing genetic variation. So there's a lot of just mutations and stuff which are in your genome which aren't doing anything. They're not bad, they're not good, they're just hanging out, but in a specific context they could be really good or really bad, depending on what your environment changes, and or your some kind of factor. So that's also an area in which variation can suddenly become useful.
And it's interesting for that, if I may, because that jogs my memory of so you can have things that are neutral, but when you can have things called evolutionary mismatches, where in the past you evolutionary capacitance. That's a cool. Is that a term, or if it's not, it should be.
I mean, it probably is. I mean, that's the whole literature on what is termed phenotypic plasticity,
right?
And the genetics associated with a trait being plastic,
right?
In a way, is it may be negative in one instance, positive in another instance, or completely neutral, and whether or not it's the same variant that's behind this plastic trait, or when something becomes attached to those different traits and then becomes kind of fixed regardless of the environmental pressure.
Right.
So there's a whole bunch of literature on that.
Yeah. Quickly going back to the evolutionary mismatch, it's when you've adapted, and actually this is exactly what kind of happened to these birds in the beginning. So it's when you've gained adaptations to survive in a specific environment, but then you either the environment changes where you are, or you move to a new environment, and those adaptations that were originally helpful in that environment now become detrimental in the present environment. And it's kind of this genetic baggage sometimes that you take with you. And that's something that we look at, that people are looking in humans more and more and more. Especially, I just took a course on this last semester before I graduated, and we were talking about how the lack of exercise causes evolutionary mismatches in human health, because our bodies have adapted to be really active for a lot every day, and now that we're not, we get all these diseases that we didn't before, and the only way to negate them is by prescribing exercise for ourselves. So
we've ended up domesticating ourselves,
right?
Pretty radically.
So. Self-domestication is such a fascinating topic. I love it. So going back to when we kind of got a little, going back to some of the earlier things. So you wanted to look in at how, when you're trying to kind of, and correct me if when you're trying to reduce these, reduce the effect of the pathway, you're trying to see what other genes are affected or like evolved to suppress it, or the other genes that evolved to get around this?
Yeah, I mean, I think that's, I don't know if you've been technically able to show that it's part of that blunting process, the variants that we found, because that requires a bit more experimentation. But initially it was just looking at variation within a number of genes, right? Because there's technically, even though the HIF pathway and those initial kind of like hub genes, the repressors and the transcription machinery and all the downstream targets are good potential, areas for convergence, didn't necessarily mean that they would be in these systems. And so it was kind of like sequencing a bunch of different areas, and then for both high and low altitude populations to compare to each other.
Right.
And then so kind of processing all that, and then running it through programs that end up attaching a significance value to each one of those variants on how likely it is that it's associated with like your trait for the most part. And there's a number of ways you can do that, but I did them through outlier analyses. So like when you have something that's like, this is way outside the norm, that might be like real. And that's why they have all these additional testing that can be done to be like, oh, that's just variation that's random. And so it does not real. But if all these other hallmarks, it shows that it's an actual real thing that's associated with it. And so I did this. That was the podcast talk that I
still need to listen to that.
It was about that paper where I wasn't expecting anything, because like I said, it didn't have to be on the HIF pathway. And it could have been at any point in those genes, because I had a whole host of things up and down the pathway, right? And it ended up being that in 2 of the species, not the third, that they converge not only on the HIF pathway, but the same genes, the exact same genes.
Cool.
And it was the transcription factor alpha dimer and the repression machinery EGLN. So that was for both of them. And so that's really unusual in a way. I wasn't expecting that, but it also kind of recapitulated some previous work for both humans and other species that also kind of found the same members to be consistently converged on by selection in different systems, to affecting a change. And then from there, I tried to see where it was in the gene, because you can have what are, introns and exons. You can have the intronic regions, which are not actually produced. They're not involved in producing a protein. And you have exonic regions that are involved, or the coding sequence are involved in producing a protein later on. So one is a little bit more meaningful in terms of when you find variation in there, because then you can see if it changes an amino acid in the protein. So what I did is I looked to see if those changes, where they were, and they were in the same exon, not the exact same place, but they were in the same exon. So meaning that they were actually in the coding region in part of the protein. And they produced a change in the amino acid. And they did. Yeah. So they're called non-synonymous. So non-synonymous, like not the same. So that's basically what non-syn means. And that exon, because you can have, I don't know, and maybe you guys got into it at some point talking about genome evolution, and introns and exons, but just because something is exon 12 in one doesn't mean it's exon 12 in another species, right? Because you can have shuffling of exons. But so I tested it. I looked to see how conserved. So how if exon 12 is exon 12 in like humans or other mammals kind of things, so that I can make an extrapolation. And it was. So I looked through the literature to see if there was any evidence that variation found in exon 12 produced like a functional like result. Cool. Because I didn't have the ability to do that, like my system, perturb them in the same way. And so ended up, there's literature that has shown that exon 12 in HIF alpha or EPAS, endothelial pass containing protein. So it's involved in the endothelium and blood vessels. It that particular exon is associated with like pulmonary hypertension in cattle that they have in like Colorado.
Really?
They have there. And so it produces some sort of result for them, although negative, as well as there are populations of people not at high altitude that are called familial erythrocytosis or polycythemia. It's just a fancy way of there's like you have a lot of blood cells in your vessels, which is not great for a long time, because it causes a lot of pressure and other issues. And so we know that hinted to me that variation in that axon could actually affect a change, and most of the time related to blood production. And so, you know, that's something to probe a bit in the future. But that showed like there is a level of, you know, convergence that's very the predictability of evolution is also very similar between these two distinct species in ducks, as well as across species, you know, in humans that also had the same genes that were selected upon, though in a slightly different fashion. It's the repression machinery that had variation in the coding regions versus the other one. So.
Gotcha.
Same but different.
Same but different. Yeah. So another quick thing. So when we talk about introns and exons, what happens when you? And I'm just going to try to make this quick. What happens when you initially code DNA? You're going to have, if this is the mRNA strand, you're going to have pieces that get read and other pieces that get cut. And the protein like knows which ones to cut. So like, and it's exons that get that are saved, right? Introns get cut, right? So
and you can have different combinations of the exons. They're not all exons sometimes are expressed. You have different isoforms of the same one. So it's more complicated, but yeah, ultimately
it's always more complicated. So quickly here, I have E's and I's for exons and introns. And so when she's talking about exon 12, because they're kind of like the order in the mRNA strand when they were initially read. Is that correct? So like you can have exon 1 and exon 2, and you have these 4 exons. So what happens when they get cut is that all the introns get just tossed, and then you have some combination, usually in like the order that they are like read in. I hope that's a right statement. I feel like, I kind of just talked out of my ass there. And so then this kind of completed exon script then gets read as a protein. So what she found is that like exon 4 here, like it wasn't the exact same part of the DNA, but this like specific exon had a mutation in it in both species that altered the protein and altered the structure and like of the protein and changed how blood vessels are expressed, right?
Yeah, I mean that's that's what I was trying to extrapolate from the results that I found and what had been found previously that they knew caused these issues like downstream. So like obviously the way I talked about it, those examples are negative.
Which negative
still it's so there's it just means that there's function exactly how that it's possible maybe it was similarly negative early on when they like got those variants, and that they have other things that come in that compensate potentially for those things. So it's kind of trying to pick it all apart. It very rarely is it's very rarely a silver bullet explanation for things, especially with a pressure like oxygen, which is like a full body experience.
Everything needs it. And so
except for when you don't need the HIF pathway. That's the other story.
So now because you didn't want to kill ducks to kind of figure out these pathways, how did you how did you switch what did you switch to, and how did you adjust to this? How did you adapt?
So when was applying for and looking for postdoc positions, I decided to go back to invertebrates, so like insects and things that don't have a spine, invertebrates. And also I figured, that had more resources available to kind of doing, like I mentioned, the in vivo, like in the system perturbation. So doing things like gene knockouts or whatever, like with CRISPR. So I moved into a system at Oregon State that are intertidal copepods. They're little crustaceans, and they had all these more of these resources that had come up for them, and they had traditionally been used as a model for environmental stressors, but mostly for like temperature stress and salinity. And so I was kind of coming at it like, well, I still want to work on hypoxia, but obviously I'm going from terrestrial hypoxia to aquatic hypoxia.
Right.
So I kind of wanted that through line. And of course, like, even because I'd spent so long on the HIF pathway to have several papers published on it.
Right.
Actually, like when I got into the lab, like within the first couple of weeks, I just wanted to like look through the genome and all sorts of other things to like keep an eye out for my faves,
right?
Like my babes, to know where they were and kind of thing. Like just as they're probably going to be important. I'm going to embark on working on hypoxia in the system,
right?
And there hadn't been anything done about it before. And in doing that initial kind of like poking around, I wasn't finding anything, which now could have been, human error, right? Like that the genome wasn't a symbol. It's like more genomes are always a work in progress, and maybe like it was just missing because it wasn't sequenced or annotated or whatever, you know what I mean? And so I did a lot of work trying my best to find it, because it was a little bit of a bummer that I understood so much about, had gotten to know this pathway and what it was doing.
And it just didn't exist.
And it didn't seem like it existed. And that in and of itself was super unusual. Very unusual. And because I had even done work that had shown previously across like basically all multicellular animals that they had a HIF pathway, right? Anything that uses oxygen, you'd think it kind of like, this seems it's going to be, there's going to be pressure to keep it,
right?
Because it occupies this really important role.
Right.
And so I found there had been only one other animal, which at the time they didn't have it. It was the tardigrade, your water bear. Yeah. So like they're weird. They could send them out in space. Like I think they just didn't. They were focusing on other oddities about that.
Right.
And so that wasn't like the big story for them.
Right.
And so this was the first time outside of something like that, which had shown that there wasn't a HIF pathway, and this is a lot more like interior into like multicellular animals, and it had not been identified. And so I did some work to try and figure out potentially what the what they were doing instead, and it answered some questions and brought up way more questions. So basically like a couple of years ago, the three men that had discovered the HIF pathway and clarified it, they won a Nobel Prize for it. So it was like,
really?
We understand that that's how this works. And so now there's all these question marks about that. And clearly it has to do with, we're looking through the retention of the HIF pathway kind of from our own human mammal lens, right? But invertebrates, especially like aquatic even versus terrestrial ones, have different needs physiologically, like for oxygen. And so I think what we're seeing there is kind of like this mismatch, is like we're surprised that there's that they're missing a HIF pathway, but they're certainly not surprised that they're missing one. So I did a lot of also that kind of changed my course, right? I wanted to go in and do all these kind of more molecular things to them in a different route, but in discovering this thing kind of unexpectedly shifted the way in which I was addressing them, which is, it happens. And, you're kind of, like I said, sometimes in textbooks and other things, you're kind of led to believe it's a super linear path. And for most people, it really isn't. And they didn't have that idea of what they were doing until they got there.
That's me.
So for them, so so I also looked to see if they were the weirdos, right? Or maybe this one's just weird.
Or like an exception. Like when they found venom in Komodo dragons, they're like, that's super fucking weird. And then they've started looking at all these other iguanids and stuff, and they're like, oh, they all have venom.
Well, it's not so weird. Yeah, I mean, it's, we're still constantly trying to, like, we still have to reframe it as, like, it all completely depends on context, right? And also, the more that we're looking at all these other things, will define just how weird or not something is.
Right.
But, so I tried to answer the question as to whether or not their loss was idiosyncratic or like their own kind of like strange thing that happened, or if it meant that we just needed, we just weren't looking in crustaceans, right? There's also not a lot of resources in crustaceans because there's certain fields tend to adopt other portions of the phylogenetic tree for their own doing. So a lot of like crustaceans and things mostly, ecology or behavior. And so there's not, that means there's not many people like creating genomes or transcriptomes for them. And so, or not looking for a specific question in the same way. So I just used a whole bunch of data that had already been put up. So I didn't generate any of that. So I like data mining. So I data mined all the sequence that was available on public servers because it's all funded by the NSF and other things.
Right. Congrats on getting funded by NSF. That shit's hard to do.
Oh, thanks. It is. It's a little bit of a bummer that I didn't get it as a faculty member, which was ironic. This is a little bit of a gripe for me. I went on faculty interviews and I had some old curmudgeons try and tell me my work wasn't fundable, but clearly it was very fundable. So what do they know?
I know, right?
So it's a little bummer, but so I looked further into all the different groups of crustaceans, which also includes barnacles. I think we forget.
Oh, right, yeah.
They're just very derived and weird on their own right.
Right.
So I looked to see whether or not they're like what the layout of loss was across all of those, and I found that barnacles lost it independently. That nobody had found before, and that there was an explicit point probably within copepods, which included other copepod groups besides T group, that had also lost it. Whoa. And copepods are a huge
family
biomass,
right?
In a lot of
they make up plankton, right?
In the ocean. So yeah, a lot of plankton. So there's a lot that we depend on them in a way, and we clearly don't really know much. And there's this thought that it was also I worked into because there's natural hypoxia zones in aquatic systems
caused by jellyfish.
Yeah. I mean, there's natural areas in which there's not a lot of oxygen, right? And that they are seasonal sometimes, but because of climate change and pollution, now some of those hypoxic zones are sticking around for a lot longer,
right?
And they're more hypoxic and they're bigger, and so we don't really know how that's going to affect. The bulk of things living
probably
there
not
that
good.
Depended on things kind of staying the same for so long. So I think that learning more about them, it might give us an insight into their ability or not to respond. So that was my work I did at Oregon State, and I'm still collaborating with them. Obviously, like me being at these places as a postdoc kind of depends on money, just like most things. And,
of course.
And my money ran out. I brought in my own money, and then so then I had to move, and so now that's how I ended up at University of Utah, the Human Genetics Department. Also working with high altitude mammals, but on a different scale.
Awesome.
So
that's really cool. So for everyone listening in, why intertidal species? Why that specifically for model switch for low oxygen?
Well, it was more, it was kind of a combination of things. It was mostly like I wouldn't have felt bad killing them. They're still cute. I still feel bad killing them. But at the time, like I mentioned, they had all these resources, but also they had already they had proven their role as a model for other environmental stressors. Like I said, most of the work that had been done on them was their ability to respond to temperature changes, because it's kind of like easy pickings. You can obviously change temperature easier than you can change, in the lab, change the oxygen availability in water. So, but in these intertidal, they're intertidal in the sense that they're a little bit more segregated than like other intertidal species, right? When you think intertidal, you think the ones that are just like crawling around when the
sand goes out.
When the tide goes out. But in reality, so Tigriopus as a genus, they have other species that live in like the Scandinavian area and like Scotland and even in Japan. So they and this particular species is all along the west coast, and they tend to be around areas where there's a lot of porous volcanic rock.
Right.
And so they live in these what they're called ephemeral, meaning that they can kind of come and go potentially, pools that aren't actually connected to like the water in any way. So they only get water that comes in through like spray. So they're very like, so you have these like populations you have these populations which are like not that far apart physically, but through genetic measurements basically are similar to completely different like species.
Really?
And so yeah, they're still within the same species. There's this oddity in terms of that. And so
speciation definition is always weird.
Yeah.
Yeah. And has this been found in like deep sea species? I don't know what oxygen levels are like way down there.
Yeah, I mean, I'm sure they're dealing with a lot, pressure included, and lack of light and so on. I actually don't know like what the amount of kind of plankton or copepod species are down further down, but obviously I think you can kind of use these guys as a potential proxy for them, because they're a lot harder to gain access to and even keep.
Oh, that's true.
If they're in the same, especially if they're in the same kind of like larger group, they're called the Hypoxia. And then
that's
there's
a
also
cool
the
name.
So and they have species and whatnot that live in all sorts of places, marine, freshwater, even some that literally just like live in like on tree bark in like little pools of water. So they're like all over the ding place. You probably wouldn't even know. And so they're, and clearly this loss of this pathway, which we think of as like,
vital
shocking,
to all life.
That we thought that everything had it. Clearly it's like a natural experiment. Instead of us going in and knocking that gene out and seeing what happens, which would most likely, the whole thing would kind of collapse because it's used to something being there. That was taken out a long time ago for one reason or another. And they've had either workarounds or it just was never really important in the first place for them, given their needs. And
they were good enough to not
unravel that question is like just a different kind of like, just a different frame of mind that you have to think about.
Totally.
Yeah.
Oh man, we have really talked a ton. I really appreciate it. There were some people asking about if they could ask questions themselves. I wonder if there were some in general, or maybe stream suggestion. Oh, it was other people just putting stuff up. Guys, if hopefully you're still there. This was such a fascinating conversation. I'm so glad that we did this.
I'm glad. I mean, I'm always like, you spend so long thinking about something and doing research. Sometimes you wonder if other people are going to think it's cool, or if you just kind of brainwashed yourself into like continuing to work on it and think it was neat. And also, I do a lot of outreach and broaden participation too. And I also don't want to ever get to a point where I can't really discuss these things more generally. I hope I did a good job kind of conveying some really high level concepts. It's really difficult, even with other scientists and even other geneticists that aren't thinking about it. So
I totally follow, like, because definitely when I was trying to do research and stuff, it was like I couldn't really, like, there were definitely parts I just couldn't wrap my head around. But like talking to you really was like, it made things super clear. I totally followed your pathway to where you are now. And it's such a fascinating experience.
Yeah, and my experience in terms of how I've gotten, obviously it's not universal, right? Not everybody's the same, but I think through the way that we kind of discuss scientists and how you get there and what you assume they're like and what they're doing, we're also just completely normal, weird people. And it's like, how has anybody gotten to the point that they're at, right? Like they
Right.
followed some bizarre route and, you kind of just, ended up someplace. And so, and like I said, I didn't come from a family that did science or,
Right.
My whole family is like art oriented. I'm the outlier for sure,
Right.
but, and I'm not, also like the model for like what most people think about for a scientist, but it is like, we're just a huge group of weirdos, just like any other. And I love doing things outside of science too. Like, so, I'm a big, play D&D with like our group of
the D&D family
friends.
continues.
My husband runs as he's the dungeon master, I paint the minis.
Oh, that's so cool.
I've been watching Critical Role for, however long they've been on since I discovered them in my PhD. And then I love science fiction and reading and fantasy.
Oh, if you like fantasy
games and
right.
Yeah, so, and I'm also a girly girl too, so like, like I like all sorts of stuff just like with anyone else. Yeah. So if anything, I hope if people take away from this, if it was too much, that
it was perfect.
I'm not like, I'm just a normal
I'm just a normal person.
I'm like a lot of people. I mean, I'm like a lot of other scientists. So I'm dumb in a lot of areas and I'm smart in a lot of areas. Like everyone has something that they're kind of an expert in. Right. So, a lot of science gets kind of shit on, I think, which is a little sad.
Yeah. It causes a lot of mistrust in science because like people who don't understand it are like, oh, the libs are brainwashing all the kids, and it's like we're just like crazy scientists that don't want bad
and it's also a misunderstanding about how money, how money gets kind of
and how
allocated
research is done.
Too. Like you don't get a blank check. Like when grants get divvied out, 50% of it goes to the university first of all.
Yes, that's true.
So we don't get to see it, and then it goes into an account, which then goes literally just to pay people. Like just to pay for stipends, for students, for undergrads, like not even for advisors. Like they get paid by the university. The only money they take out of it is during the summer because they don't get paid during the summer.
Right.
So it's like, it basically is it's paying people for jobs, and there's stuff that gets spent on other things like, other materials, but a bulk of it is, it's not just people,
blowing it all on big part
stuff. Like, yeah, so there's a misunderstanding about that, and there's steep competition to get it.
So
the funding
much
rate
competition.
Is like, 3 or 5%. There's a lot of people fighting for money, and we don't really invest as much as other groups. Yeah. So other other countries and things.
Right.
So hopefully, if anybody's taken anything from that, it's like just the human element.
Right. That's awesome.
So. And, feel free to, if anybody if there's like papers that you guys ever want to have access to that are not open access in open access journals or whatever, just like you can talk about it on Discord or whatever, and I can upload the PDF. Like
totally. That's awesome.
Like I have access to those.
Sci-Hub is the way to go though. Sci-Hub is the way to go. You get everything free access.
Sorry.
It cut out for a second.
Sorry, you cut out.
Sci-Hub is like one of my professors showed me it.
Yeah. Oh, yeah. That's also a place too.
Such a good place.
I've done that sometimes. But if there was, if for some reason you couldn't even find it there, or if I wouldn't mind doing that. But especially too if it's like my stuff. If you wanted to read about my things if something has possessed you to read my work, then I have no problem providing you with copies of that if anybody wants to talk about it or whatever. So yeah.
If you're still willing, if your voice is still strong and the spirit is still willing, we have a number of questions from the audience.
Sure, and I'll see if I can answer them.
Yeah. So Pepper Steinson comes in saying, people feeding ducks, does it harm their do harm to their behavior? Are they too used to being close to humans? Depending too much on humans, what can we make them more wild again?
Well, I mean, there's a lot of things which I think obviously feeding stuff. I think in general, the consensus is feeding wild animals is like
Bad.
Probably a big no. Obviously we do feed them for, bird seed, we put birds out, you're trying to encourage things that are migrating. I think the biggest issue, and I would see signs about this too, like, is what you feed them. If you're going to feed like, especially ducks, if we're talking about that, feeding them white bread is, even for people, is not a rich source of nutrients. So they end up having developmental problems. I think it's called like angel wing. So they can't, like, they become dependent on humans for food in a way, but also it hurts them, like, physically. So if you're going to feed them anything, like, if you're going to feed them anything, you feed them stuff that they're, like, generally used to eating, which they're vegetarian in a way. They're eating lots of algae or plant material.
Or
if
tadpoles.
you're going to feed them bread, like whole wheat or something, right? So I think there's a fine line there in terms of, like, the general rule should be, like, you shouldn't be feeding wild animals because you don't want them to become dependent on you and then invade your space. We're in their space anyway, but become kind of a problem. So yeah, there's those two kind of elements to deal with.
Absolutely.
There's no right answer, I don't think, in reality.
I think that was a good answer. That you should avoid it, but if you do feed, make sure it's not something that's going to hurt them.
Yeah, yeah.
The next question, by What Did Corgis Ever Do to You? Which I love that full title.
What about corgis?
He's well, that's the title. Is this What Did Corgis Ever Do to You? So the question he had is, did you have any formative experience that made you want to pursue a career in biology? So I guess like a teacher or a class or something.
I've always been interested in some sort of science. It's kind of hop, skipped, and jump around. I'm probably like a lot of kids that liked dinosaurs, and then once I found out you could study dinosaurs potentially, like, I didn't really know what that meant.
Paleontology always brings it into science.
No, it really does, and I loved it, and I was always really curious. And my mom, we had lots of books and things, and she was also like if we don't know, let's look it up, and we'd go to the library. So I was always very curious and looking things up, and I will be the first to tell you if I don't know something, let's look it up.
Totally.
So it was more of just a general interest in those questions, and then at some point I discovered like genetics, Punnett squares and the whole thing, and then like it kind of just, so I've always really liked science. In terms of kind of to getting to biology, it was more, I made that distinct, connection in high school, and it wasn't anybody specifically. Like, I had already kind of made up my mind that I wanted to do biology, not in a medical sense, because that's just not like my personality. Like, there's a lot of people I knew that were like, they want to be doctors because their parents were doctors, or they wanted to make money, or they liked helping people or something, right? Like, I liked helping people, but I didn't really want to,
animals were cooler. You just liked them more.
When they have the only time people, when they come to you, is going to have a problem. And that's like, that's a lot. And so for me, it was more just, I just like studying like nature, and there's so many fascinating questions. And like I said, my grandmother was a big birder, and she got my parents into it. So I was looking at things and watching animals. And so it was like trying to connect those two things. So it was, like I was never thinking like, I'm going to be a professor. I didn't know what that was. A lot of people, even at this stage, don't really know what that is, even though they're trying to become one. It's kind of this weird concept. So it's just more, it literally was kind of like, I'm very like, follow your heart. Like there's a lot of things I'm sure we've all done that were inadvisable in a way, but a lot of things that I've done is when people have told me like, oh, that's not going to work, or, you shouldn't do that or whatever. Like, I'm very driven, as like some people driven by spite, right? Like, I'll make it work.
Rage.
Like I can do it, and if And if I don't do it, I'll be upset that I didn't try.
Right.
You know, and so like I try and leave a lot of stuff on the table.
Totally.
So that I can't be disappointed in myself. So
Awesome.
so yeah, like I've always been interested in science and those type of questions and biology in general. In terms of like I've just kind of taken it one step at a time as I've gotten older, and once I find out more about opportunities available and whatnot. So yeah. It's
that's awesome.
I knew some people that really had this, like they were going to do this and this, and like they've always kept with that or whatever, and which is admirable. But there's a lot of things that happen to you and change, your interests change and your life circumstances change, and so I've evolved that way.
That's awesome. No, I think that's the right way to do it, is to be adaptive and just keep following what you like, and you'll get somewhere eventually. Another question: what is the rarest duck species? Do you know that? I don't know.
Oh God, I'm sure there's some really. I'm trying to think. I bet there's a few duck species on very discrete islands, which have very limited populations. Like as much as I technically studied ducks, I don't really know like a lot about them. And there's so many fun, color combinations and things, and that Kevin had worked on. That there's the torrent duck, which actually dives at high altitude, and they have these really cool orange beaks and are black and white and really interesting. And I joke that I had the duckiest like ducks. They just were like brown. They didn't really, they just look like any other duck.
There's the lamest ducks.
I'm not really much of a duck. I'm a really bad duck person, and especially bad as a birder. Like I don't really know a lot of the genus species. I know their common names, and I can pull some out based on their sounds, but usually that's only because I've been living someplace so long I get used to it. So in terms of very specific ones, I can't think, but there's unfortunately so many populations of birds that are island endemics, which are under have historically been, under attack by, invasive species of things that get brought in, like rats and snakes.
Right. Right. And so the next question by Renrar Emit. What's your favorite bird? If you have one, or if you can't pick one, top 3.
Okay.
Yeah. I'll wait till the next thing. I'll wait till you answer this one.
Okay. So I was always fascinated. So I grew up in Houston, and I have always loved mockingbirds, and I really have missed them because they haven't been in the places I've lived in recently. They're just so sassy. I'm big into sassy birds, because my other favorite is Carolina wrens, like just wrens.
Oh yeah.
They're like little balls, and they have like a tail, and then they're just like, they just give you the stink eye and whatever. So I love wrens and mockingbirds. In terms of like, let's see if there's another third one, I'm trying to think. So I think mostly the mockingbird one is like a comfort. There's something comfortable about them.
Right.
And I don't know. I love all their little mannerisms. Let's see some other, and I just saw them again because I hadn't seen them in a long time. I really do like their kinglets, little ruby crowned kinglets. They're so small. And they have this little, like, the males have this little tiny red thing that you only see when they get like irritated. They pop up and it's like red, and they're just little balls. I don't know. I like, of course you can't really beat out like chickadees and titmice and all those things, but I think those are like my top kind of 3. Nothing particularly flashy, just there's something comforting about a mockingbird and their mannerisms and like all their songs, and all the stuff that, their ability to kind of mimic things, and yeah.
Awesome.
Yeah. That's for me.
And the second question is, have you read this person called Richard Prum? Have you ever heard of this person? Prum?
Uh-huh.
What do you...
I met him.
Oh, Cool. I do not know who this person is. What is your opinion on his theories? I don't know what they are.
Is it about the like sexual ornamentation, like sexual selection? He's got it under a number of things, if I'm remembering that person correctly. He's from Yale.
If he's done a bunch of those theories, damn. Let's see.
He's got a lot. The last one that he was trying, he came for like a series at Miami when I was a PhD student, and I feel like he was trying to. I don't even remember what it was. I was just knee deep in his in my own work, and so I was kind of just like, I don't really want to be here. I want to work on my own stuff. I gotta get out here. Yeah.
The biology of beauty. That's what he says.
Yeah, that's what I remember. That was the latest one at that point, which he was trying to suggest. I don't remember what the details of it were. It seemed mostly like theoretical, right? I mean, a lot of these, there's a number of men that are like that they'll have all these kind of like, they come up with these ideas, and then it kind of falls on either them or other people to test those theories like out in systems and whatnot. So I don't know how much has actually been done to address that, though I'm assuming his lab is trying to find potential evidence to support or not his ideas about ornamentation and how beauty in the eye of whatever beholder you're talking about is kind of evolved separately. So
interesting.
Yeah, in terms of specifics, I haven't really kept up with the sexual selection field at all anymore. I used to really like that stuff a long time ago, but
gotcha.
Yeah, so I'm sorry, it's not a satisfying answer. So I guess the answer is I don't really have a particular opinion, though. Though I've heard him give talks on it. So he seemed like a very nice man.
Sweet. How would you like to further your research? That's another question.
Oh gosh. Well, I think the first thing starts with me having a faculty position. So, it always it seems like at my stage, I'm kind of doing what I'm supposed to. So my research is progressing in a way. And thankfully, we just got an influx of money through the NSF grant that I'm called a co-PI on. I can't technically be a PI or principal investigator because I don't have a permanent job. Right. But I co-wrote it, and it's based on all the work I did as a NSF fellow. So we're going to eventually try and do all the experiments that we had laid out there, which is great, and we'll maybe uncover new and interesting things to embark on later. I come up with another series of potential projects using zebrafish, a more established model system. We have a big facility here at University of Utah. And I wrote what is called my K99. It's an NIH, National Institute of Health, big grant where it has a mentored and non-mentored phase. So we'll see if I get that in a couple months. But so I think my first inclination was correct in terms of being able to actually have some control and direction over my own research interests. It requires me having a lab and being a faculty member somewhere. Because right now I'm still kind of at the whim per se of the people above me. Right. The availability of money, my availability of me bringing in money to do exactly what I want. Because there's a lot of people that are in my type of position which have not been as able to pursue their own questions because they haven't been able to bring their money and own money in to do stuff. And so I've been very thankful. I've been very lucky that I've been able to kind of embark on the things that I'm interested in. They've uncovered interesting things because there's also the potential that what you do is not particularly groundbreaking, but it's still a result and still something we didn't know. Which is also valid too. Not everything has to be a science or nature paper. So, I think if in terms of a one-liner for answering that question, that would be the surefire way to me to actually have more direction over my own research interests. Because right now I'm also working on other people's stuff. Besides mine. So.
Pepper Steinson has a silly question. Who would win in a fight?
Okay.
One mute swan or five mallards?
Oh gosh. I mean, isn't that kind of age-old question? Like, you know, what is it, like a horse-sized duck or a bunch of duck-sized horses
Oh,
or something?
right.
You know, I I feel like swans. I haven't had as particular interactions with mute swans, but I feel like they're definitely a lot more on the offensive than
They
ducks
are
are.
mean. They
So I honestly would put my money on the swans, and then the mallards just kind of chickening out, and giving up. I think
that
so.
seems I would agree with that.
Yeah.
Uh and another Renoir Emit asked, you mentioned computational techniques and data mining. Do you code?
So I'm using a number of different languages, none of which I'm incredibly proficient at. I think this is like jack of all trades, like master of none.
Every scientist has to like struggle
Yeah,
with a data
it
with
is.
stats.
And there's so many different, there's so many different ways in which you can accomplish something.
Right.
And also there's plenty of people that have already there's so many programs kind of pre-made and code pre-written that
Right.
I've kind of become okay, especially if it's on the smaller scale, kind of retooling for my own needs. So I very rarely kind of start from the ground up.
Right.
There's if you want an exercise in something, I guess you could kind of code from the ground up. But why reinvent the wheel if somebody has already kind of made something? So I kind of sit at that intersection. There are specific people specifically that their whole lab does coding to create those tools for other scientists to use. And I'm not nearly as good as they are, so I kind of leave that to them. But so I'm a little bit more familiar with Python. I have used some Perl. I'm not entirely as used to that. I'm getting to know R a bit more, but I'm a lot more familiar with like Unix and Bash scripting, but none of which I would, put necessarily money on. It's been able to get me. My abilities have been able to get me through, and I work definitely better on like it requires a task to get me to do it rather than like I'm just gonna start learning something. Right. It really is like a more of a throw me into the deep end. I learn better that way, and it gets better results for me. Obviously, everybody works differently.
Right.
So I do code some, but not as many as some people, and definitely more than others. So I sit kind of in a weird position.
Right. That's actually something that that's why part of the reason I'm getting my master's in statistics, well, I'm trying to, is because like data analysis and stuff is like difficult at times, and like you could go to a stats person, but it's always difficult to explain what you're trying to get and like what you're trying to actually study, and like and they may give they may use programs and stuff that you have no idea about that you try to justify in your paper, and it's like that I was feeling that I thought that it would be good to have someone like in a lab or in the department that knew the science that you were trying to get and could then like help working on like the statistics for that.
Yeah. And I know that some labs, depending on just how much of that they do, they actually employ like a specific like research associate in their lab that does it. Unfortunately, there's a lot of labs which, like I mentioned, that they do this bioinformatics and computational heavy thing that people will go and try and collaborate with them and then just kind of use them as a tool as well instead of like recognizing their use, right? Like it's unfortunately. So it's also important to cite programs and things that you're using if you're using other people's work. So yeah, I mean, there's a need for that. I mean, it was becoming difficult, right, in terms of just how much you're required to kind of do and the interdisciplinary nature of the work that you're doing. And it doesn't necessarily mean that you have to do everything like yourself, right? Some people are really good at one thing and, know the ins and outs of it. And so that's why collaborations with labs and other people are so important to do.
Totally.
Yeah, so I'm really excited to be in a position now that's that's a little bit I'm able to kind of rope more people in to help, because I was in I was in Kevin's lab, and it was very like me doing everything, which on one hand was kind of good as a PhD student, because I was really getting in there and like really doing it all. But at the same time, I would see people that, they would end up being on these papers and things like with a bunch more people. And it's like, I would hope that they had all this additional help,
Right.
where these days where, it kind of does take a village. So,
Right.
So yeah, so that kind of thing is I do code and stuff, but at the same time, I'm fully aware that there's limitations for everyone, and that a collaboration with people that are more handy with it,
Right. Totally.
are good too, so.
All right. Well, I think that's like everything well, there's probably definitely more things we could talk about, but I feel like that we've had quite the thorough conversation.
I think so.
It's been an enormous pleasure talking to you and learning all about this. I've been so excited about this whole week.
I'm glad.
And
And you got me on a day that I was primed for it, because I had to give I had an 8 a.m. seminar in hematology, which is always tough. So I was like, my brain was charged immediately. And then I gave a seminar at Clarkson University over Zoom and talked about my stuff earlier. So I was like, the brain was already like going
primed.
on it. But awesome. I'm also glad that you were excited, and that hopefully I lived up to the hype.
Totally.
Or whatever.
Totally.
So, and hopefully other people in the chat, since I can't really see them, I closed it because it was too much time to fill, that they thought what I was talking about was even moderately interesting.
I think it was, and it seems that what everyone was saying was like, this is so cool, listening in and everything. Is there anything more you'd want to add before we wrap it up?
I don't know. Not necessarily, other than just, believe in science.
Believe in science.
Believe in science.
Believe in science, but at least look at the statistics and make sure they're good.
Yeah. Well, yeah, scientists can be wrong too, right? It's not all just one data point. It's a bunch of data points that kind of help you. So it's a complicated process, and we're not scheming.
I kind of want to grow the mustache, the curl as I ski my way, but. I didn't have any particular outro or whatever, but if at the very least, like I said, if you guys, if anything sticks, is that, I'm just like a regular person, and now you can say, like a scientist, and a
always
doctor.
Reach out. Anybody, just like I'm on the Discord if anyone wants to talk about anything, or even non-science related, but just like life, if you wanted more of a story about my experience as a PhD student or whatever, which is like a whole ass thing.
Definitely.
And yeah, just, feel free to send me a chat.
Yeah, that all sounds great. Allie, it's been awesome. I'm probably still going to be streaming, so just join the chat again.
Sure.
And we'll just...
I probably need to start thinking about dinner soon.
Yeah, me too.
I haven't really eaten anything today since I've just been busy. I had a little bit.
I brought myself pretzels, but I haven't eaten any because I've been so...
I have, bunny cookies
right.
I was eating earlier, which I shouldn't have it within arm's reach, because I just keep stuff with them. So
totally. All right. Well, everyone, I hope you learned a bunch. I learned a bunch. The stream was going to keep going, but
okay.
Thank you so much for your time. It's been a great pleasure.
Yeah, no problem. I hope everybody has a nice Friday and a nice weekend, and I'll log out of the Discord and hopefully record.